All right, welcome everyone, and thank you for joining us. I'm Dr. Penny Dacks, AFTD's Senior Director of Scientific Initiatives and the President of the FTD Disorders Registry. Today we will be discussing the Alector Therapeutics Phase 3 clinical trial for Latozinemab and the general state of development for treatments to slow or prevent progression of FTD. I'm joined by Dr. Carmela Tartaglia, the Chair-elect of AFTD's Medical Advisory Council, professor at the University of Toronto, and cognitive behavioral neurologist at the UHN Memory Clinic. I'm also joined here today by Jari and Heidi, two community members and advocates whose families have been impacted by FTD caused by variants in the progranulin gene. Welcome, Carmela, Jari, and Heidi.
Hi.
And before we get started, I have just a few quick announcements. So first, we have muted our audience to minimize background noise. AFTD staff are working behind the scenes and are available to help you if you encounter a technical issue. Just let us know by writing a message into the Q&A box that's on that Zoom interface. Second, we've already received questions from the community that we've structured this agenda, so to speak, to answer today. So to submit additional questions, use that same Q&A box to submit your question. If you want to submit a question anonymously, you can do that too. Just click the box to the left of the send button. Please submit your question at any time. You don't need to wait until the end of the webinar, and we'll get to as many questions as we can. Finally, this town hall will be recorded and archived for later viewing on AFTD's YouTube page and in the webinars section of our website. So our goal with this webinar is to, again, answer some questions about this trial, as well as what it means for the path to effective treatments. If anyone's hoping for a really nitty-gritty discussion on the science behind this clinical trial and the results, you might be disappointed because all we know so far is what's in the press release. But there's still a lot of questions that people have, and we didn't want to wait to be able to have this conversation and answer what we could. So first, a little bit of background here, because some folks won't be familiar with this. So the Alector clinical trial on Latozinemab was enrolling people specifically with an FTD progranulin or FTD-GRN genetic variant. What we mean by that is that with their genetic status, they have a major reduction in how much their body creates of the progranulin protein, particularly in their brain. So that is linked to many different types of downstream consequences, including neuronal cell death and microglial dysfunction, things involving neuroinflammation. It can cause, I'm not going to go into, again, the nitty-gritty of all these pieces, but the bottom line is that it really raises the risk for developing FTD. Now, there are some modifiers, so it's not every single person who is going to get FTD, but it does significantly raise the risk. And so, again, all of this is related to decreased progranulin levels. That's what the gene is doing. So, or that's what the genetic variant is doing. So this treatment was designed to increase the levels of progranulin in the brain by inhibiting the breakdown of progranulin. Thank you. So the clinical trial by Alector was the first ever phase 3 clinical trial for this type of FTD. It was a long trial. It took 3 years for Alector to recruit participants who were then treated for 96 weeks. 2 weeks ago, Alector announced that the treatment, while apparently safe based on the results of the trial and effective at increasing progranulin as measured in the blood, it did not appear to stop or slow disease progression as measured by a clinical measure of disease severity, as well as by biomarkers of disease progression. So this analysis was really based on the people with an active FTD diagnosis. The trial also included a small number of people who are at risk of developing FTD in the future, and their results are still being analyzed. It was not included in what was shared with the community in that press release. So overall, based on the results, however, Alector and their partners at GSK have discontinued development of this drug for FTD. Now, let's be clear, this is a huge emotional hit for a lot of people in the FTD community, pretty much everyone in the FTD community. And we're going to talk more about that in a minute. I really want to emphasize, however, that this trial is a huge step forward to effective treatments for FTD. Every trial is an experiment. It's a test if a treatment is safe and effective. And if we knew that it worked, it would be unethical to do the trial. And as we think about how urgently treatments are needed for FTD, how we will get there is by having a lot of shots on goal, by trying many different possible treatments, by learning from every trial to guide the next one. This was the first ever phase 3 clinical trial for FTD progranulin, with an unprecedented number of clinical trial sites for an FTD trial. So before we get started, I really wanted to start with a thank you. On behalf of AFTD, I want to thank every person that was involved in this trial. The many people who participated, from persons diagnosed, care partners, at-risk individuals, all the family members and community members that supported them, thank you. To the scientific community, whether the Alector staff, their GSK partners, to the many, many investigators at trial sites who were working face-to-face with the families, thank you. Heidi, you were a participant in the trial in the at-risk arm for people not living with active FTD.
Mhm.
Can you tell us a bit about what participating in this trial was like for you and how you felt on receiving the news?
It's been participating in the trial is kind of like for a while, it was like a roller coaster at the first, finding out that you have the gene that affected both my mom and my grandmother, because both of them had FTD. And then knowing about the gene and then going on the trial gave you that hope. And it also gave you that sense of purpose too, like that everything, like there's a reason why you got the genetic mutation. I felt like there was a reason why the genetic mutation was to do the trial. And it gave you the hope that you're not going to get the disease your mom had, or that my mom had. And when I found out about the ending of it, I have to admit it was emotional. Like I did have to have phone calls with my family, all my family members that night. And my family members reminded me that there still is hope, but you have that first, wait a second, you're starting back at square one, fear. What's gonna, and I made the decision when I found out about the clinical trial to get tested and find out about my genetics. But then also you start to feel better because you realize after talking to you guys and speaking with other people and reading up about it, it still was a huge progress in FTD. So, and then another thing too is I started to become like UPenn started to become a community for me where I looked forward to seeing them once a month. And I mean, I'll see them again for the follow-up visit, but you kind of didn't have that goodbye. And then also I had did a, I was a fake person during it. So it's kind of like I let go of my fake name that I had during this study. So I even told my siblings farewell to my fake name.
Yeah.
So
Well, thank you for participating. So before we discuss the trial further, we wanted to talk briefly about what's happening in FTD clinical trials and what are some of the other trials happening. So the slide that you're going to see in a second is that for the active clinical trials that are specific to FTD progranulin. So there are, oops, here we go. So Alector was one trial of many that are actively in development, and these are just the ones that are in clinical development for FTD progranulin. So Alector was the farthest ahead for sure. They were the only ones in phase 3. But as you can see on the slide, there's quite a few other trials that are each developing their own approach to these treatments. I also want to mention that there's a lot of other treatments that are not for FTD progranulin, but for other types of FTD. Not as many, but there are a lot of treatments that are in development, particularly in the preclinical stages that haven't yet reached clinical trials. So Carmela, can you tell us a little bit more, expand on this about the clinical trial landscape, particularly for FTD progranulin? One of the questions we've also been hearing a lot is to understand how the Latozinemab results, the Alector results, what does it tell us about these other trials? And if the Latozinemab raised progranulin levels in the plasma, but didn't help people, what does that tell us about these other treatments that are in development that also raise progranulin levels, but in different ways?
Yeah, I think that's a great question and an obvious one. But I would remind people of the amyloid space. So in Alzheimer's disease, there have been probably over 100 trials targeting amyloid. The thing is that every trial, the product is slightly different. and does its job in a slightly different way. And it took many, many, many years and many, many failed trials to finally have some that actually are doing what they said they were going to do. That, you know, going after progranulin is kind of like a low-hanging fruit, right? Like if people don't have enough progranulin, you're going to try to find a way to raise it. And you think, yeah, that should help. But you have to understand, in the Alector trial, the progranulin was raised in the plasma. So we actually don't know if it was raised in the cerebrospinal fluid. Maybe we'll find that out. A lot of these other therapies are really trying to raise the progranulin within, like around the brain. And so many of them are more invasive than, not all of them, but many of them are more invasive than the Alector trial. They're actually, you know, injecting within the space near the brain. Some of them are gene therapy, so really trying to correct that progranulin gene. So I would say that going after progranulin is a viable, logical goal. And thus the results of this trial don't negate that. You know, one of the issues that we have when thinking about a drug is you have to get the drug to where it needs to go, and it isn't easy to do that. The brain is designed to protect itself from everything else. And so, you know, we don't really know at this point what that drug, like how much of that drug got there, and whether there was enough to do its job.
That's really helpful. Thank you. So I just want to reinforce for a moment as well that about what Carmela just said about the number of clinical trials in that amyloid space, right? They had many shots on goal to find a treatment that was going to help with amyloid pathology. And they're still in development. There's other types of treatments that are still in testing. And I think that piece, how do we get that happening in FTD? How do we have as many shots on goal as we can have so we find those right treatments that are so urgently needed? And there's something that I think is really important for everyone to understand, which is that clinical trial readiness. Part of why this Alector trial was so groundbreaking was not just because of the testing of Latozinemab and what we're learning about progranulin biology. It was also the first phase 3 clinical trial for a genetic cause of FTD. And they took, I mean, it was 3 years and an unprecedented number of clinical trial sites to find 100 participants. It was a lot. But frankly, it happened, all of this was taking place in a space where genetic testing and counseling is not standard of care in the United States and elsewhere. It was a big lift, but it was achieved. It shows the field that we can run these clinical trials, that we can, we have an active, informed patient community ready to participate in research. And that is really, really important to convincing other companies and investors that they should apply their treatment ideas here to this patient community. So again, it was a groundbreaking trial. Jari, you have been a committed FTD advocate for many, many years. You first joined the AFTD Board of Directors in 2010. For example, you've been very deeply involved with the FTD Disorders Registry. You have participated in research yourself, and I know that's just a few of the ways in which you've been driving progress in this field. Can you, thinking about that, like what you've observed over time, how would you describe the progress in the science and specifically drug development?
Well, thank you, Penny, and AFTD for hosting this town hall. And first I want to say, and I want to echo what you had said earlier, that obviously this has been a very sad finding, I think, for the FTD community, especially for the progranulin community. But that said, again, I really, really want to thank Alector for initiating and completing this study, and also thanking everybody who participated in the study, including those either with a formal diagnosis or those who are asymptomatic, plus their study partners. It was not necessarily an easy study to be in, and the study partners did so much. You know, and what I will say, like as far as drug development is concerned, I first became aware of progranulin in 2006 when my brother was diagnosed with FTD. And was identified as a progranulin carrier. When I first started looking in PubMed, which is the central database for all published studies, almost all the previous references to progranulin were in regard to cancer. And now when you go to PubMed, there are thousands of publications specifically dealing with progranulin. I think part of that is I really want to acknowledge and thank Bluefield Project to Cure FTD for a lot of the work that they've done. I mean, they started out in 2008 as a consortium for FTD research, focused on very, very basic research just to understand progranulin genetics and the basic biology of progranulin. And in 2010 is when they became a nonprofit where they wanted to do that translation to find, to take those findings of the research consortium and develop those into drugs to treat FTD. And there's just been this explosion since 2008 of understanding progranulin and the role that the mutation plays in neurodegeneration. And I would, and as disappointing as it was that Alector was not successful, we are also very lucky to have 5 other clinical trials that have been shepherded through this process. And I also want to really stress that it was this early investment in basic research that paved this path through very, very complicated landscape for there to be so many clinical trials right now in the progranulin space. And I also want to just acknowledge too, the FTD Disorders Registry. It's a phenomenal tool for facilitating research, and Bluefield was just a really, really wonderful partner in developing the FTD Disorders Registry. So there has been so much movement as far as science is concerned. And again, even though this has been really disappointing, we have never been in a better space than we are right now. Thank you.
Thank you, Jari. Heidi, people that have participated in the Alector trial can still enroll in other trials as long as they've gone through the washout period and they meet the rest of that study's criteria. Do you have any suggestions for people when they're considering participating in a trial?
Yeah, the one thing I would first suggest, make sure that you have a support group, not just a support group with people that have your family members that could have the gene, also a support group of people outside the gene. Make sure too that you do have a therapist on hand, because once you find out if you have the gene, you can't go back. You can't go back and change if you have it or not. And you also have to realize that when you find out about a genetic gene in the family, even though it's your body and you do what you want with it, it can open up a can of worms for other family members. So you do have to be sensitive towards how other family members will react, especially when it's a family that's experienced it like time and time again. Like for my example, my family was my mom, my grandmother who had it at the same time. So my family did experience 2 losses at once. So sometimes people want to kind of walk away from situations, and when it's genetic, it's always going to come back. And so you do have to make sure you're sensitive and just make sure that you have the support group. And I was very fortunate to have a group of friends that were super supportive, especially my study partners, which were both my friends from college. And I work as a contractor at NIH, so I also had NIH staff like support. And then on top of it, I also had a group of family members that were not directly affected by the gene that were supportive to me, that I could go to instead of going to the family members that could possibly have the gene, so I didn't upset them. So those are suggestions I do have for people.
Thank you. I do also, I'm going to probably repeat this a couple of times, but we won't develop effective treatments unless we have people willing to participate in research. That's just the reality. We need the community to be partners. And every family and every person needs to decide for themselves what's right for them. It does not mean that every person should feel that they have to get testing and that every person should feel that they have to go. Like, people need to make this decision for themselves. But I just truly applaud the courage of the people that participated in this study, because we will not stop this unless we are able to run the trials to test these treatments. Carmela, can you please help us understand how a drug could get to phase 3 if it doesn't appear to impact clinical symptoms of the disease?
Yeah, well, that's phase 3 is, you know, the definitive test, right? You need to have trials that are large enough and that their goal is to evaluate whether a drug is effective or not. You know, people often hear of things, you know, in media or on the internet and, you know, making claims, right? But phase 3 is where you test those claims, and a lot of drugs actually don't want to be tested, so they don't go down that road. That's the only way, and you need enough people to show that the drug is efficacious. The earlier phases are important for, you know, showing that there's target engagement, that the drug is safe and not toxic and, you know, has, you know, limited side effects. But phase 3 is really the test, the ultimate test, whether something works or not. And so, you know, we would want to be able to predict before and instead of getting to that phase, because those drug trials, especially in our space, in the neurodegenerative space, they have to be placebo controlled. So some of the people don't even get the drug, right? But that is the only way for us to really prove that something is working and is doing what it says it's going to do.
Thank you. Jari, what would you say to people who have misgivings about enrolling in the Alector trial?
Thank you, Penny. And actually, that's an interesting question that you're asking. As you know, there is another, there's a progranulin-specific organization called FTD-GRN that Wanda Smith formed here a while ago in the past few months. And there have been a number of meetings where this particular issue came up. And I think that in discussions with this, one of the things to focus on is the fact that when making a decision about which clinical trial to be in, the decision is based on the data that you have at the time when you make that decision. And I know how sad and unfortunate it was that Alector didn't meet the endpoints, but people made those decisions for very, very specific reasons. I know for some people, it had more to do with geographical considerations, that the Alector study was easier. For other people, it was more important to them that it was a study that did not require their loved one to go on an immunosuppressant, especially during COVID. And so that impacted the decision that was made. And again, all of these decisions were made at that point in time. And even though the study did not have positive findings, it is really important, again, to reiterate what you've been saying, Penny, that we have learned so much from this particular study, even though it was not successful in the end. And we can't thank those people who participated enough in the process, and especially, again, to their study partners for all the work that they did. Thank you.
Yeah. And I will mention just because we just had a question pop in about, some of the clinical trials that are active right now, some of the gene therapy ones, for example, don't have a placebo arm because they're at that earlier exploratory stage and for various reasons of the trial design. So no, not every trial currently has a placebo, and that's also we've heard from community members that that's part of why. Though certainly once you get to the full randomized, like to the phase 3, that is usually the expectation. We've had quite a few questions come in while we're chatting. Carmela, be ready. Most of them I think are going to be for you. So one question was, since asymptomatic patients were not analyzed, what if the drug is helpful for delaying the onset of symptoms? Could it be possible that although it was ineffective for the symptomatic patients, it could be effective for those without the symptoms? And Heidi, please feel free to chime in after Carmela as well, if you want, as a participant.
Yeah. So I guess that's a great question, right? And Heidi's probably asking the same question. We don't know, right? We don't know if it works for asymptomatic. One of the issues we've had with the Alzheimer's trials is that for a long time, patients who were in more advanced stages of the disease were given these drugs to target like the protein, to get that one of the bad proteins out of the brain. And what's been recognized actually at this time is that only if you're in early stages of the disease can you actually get benefit from this, like substantial benefit. So that's a great question, and I don't know that they've enrolled enough people to be able to answer that question. And some trials in the future might be directed at only people who are pre-symptomatic or asymptomatic. So an ongoing open question. I'll pass it to Heidi.
I don't really know if there's. All I know is that I've taken it probably since 2019 up until September of this year. I had a little bit of a gap during COVID, but I don't know if it actually helped. I know it stayed in your system for like a week or so, and then every month you would have to get it. It didn't really affect me too much. If you guys want to know, like my day to day, I still felt good. At the beginning, I felt a little bit flu-like, but after that, I was fine. I ended up becoming an ultra marathon runner, so it didn't really affect me. I don't know, it was the fact of finding out about the gene that made me become an ultra marathon runner. But besides that, like I don't know what the results are for me because I was always in the phase 2. I was never in phase 3. I was just in phase 2, extended.
Right.
So,
and one thing we can say is that we've heard from the Alector team that they are committed to getting those results analyzed and put out to the community. And I think, it's a testament to their committed staff at Alector that are going to want to make sure that the field really learns as much as possible from everything that was done in this trial. So if there is a signal of benefit from maybe some of the biomarkers, for example, then it's the kind of thing that could guide the next trial design. I think it's also important to understand that some of the approaches to prevention, we need tools to measure if it is indeed preventing. And one of the things that I understood is that, Alector had enrolled people based off of a biomarker thinking that would then predict who was going to develop disease. And what they were finding is that they were not as good at predicting it as they thought. So then it's hard to show if you're slowing the onset of disease if you don't know when disease is going to have onset. So it's something that the whole field is working towards. And frankly, it's part of that clinical trial readiness. Do we understand how and when the disease emerges so that we can then detect if we're slowing or stopping the progression in a quick and effective way instead of waiting for years of waiting and waiting to see if we're going to find a benefit? All right, another question. Are the people with a different genetic mutation eligible to participate in studies like this one that are more specifically focused on a different mutation? Carmela, can you take that one as well?
Yeah, so this trial and any of the trials, like most of the trials aimed at progranulin are actually only enrolling patients with progranulin mutations, which kind of makes sense. But some of the trials have opened it up because they feel like the mechanism of increasing progranulin could help other mutations. Not all of them, but some of them. So it's really on a case by case, and that makes sense, right? Like, these drugs are not benign. They can have side effects, and it has to be worthwhile for you, right? Like there has to be a potential benefit for you to enroll in a trial. And if the mechanism doesn't make sense for a different gene, then it's probably not to your advantage to be in the trial. But the companies, I think, are interested in making them as available as possible. So I think it has some of them that are, one of them at least that was aimed at progranulin has opened it up to C9 mutation carriers.
Thank you. So I think this is a question that's going to first be for Carmela, and then I invite Jari and Heidi to both chime in. So for people that are not eligible for disease-modifying trials, are there tips for handling symptoms? And I think it'd be, again, great to hear from Carmela as a clinician, and then from Jari and Heidi as community members about how families can better. I know it's a big question, so maybe just put forward your top line answers. Carmela?
Yeah, I mean, I think, we have symptomatic treatments. And so we can treat mood, we can treat some behaviors. I think, those are what we call symptomatic treatments, and they are readily available. We treat pain, we treat agitation. There's many things that we can treat. We have, fairly decent treatments for. And that category is different from what we call disease modifying. And that's, what Alector and some of these other progranulin targeted therapies, and those are disease modifying. But we have many symptoms, symptomatic treatments that can help people.
Jari and Heidi, would you like to chime in?
I think thank you, Carmela, for talking about those specific treatments. I think that every family and every situation is going to be very, very different. And that said, I think one of the best ways to learn about some of this stuff is by developing relationships with other people who have gone through this before. There's multiple support groups, both through AFTD as well as through other organizations. There are also newer organizations that have sprung up that specifically offer those types of that offer support groups or opportunities to talk about what works. And I think a lot of times, it's great to talk to other people who have gone through this before you have, to find out, like, what did you do? What was successful for you? And is that something that perhaps I can try? So thank you.
Thank you, Jari.
I would say, like Jari said, go into the support groups. Whenever my mom was first diagnosed, I started going to a support group to understand, and that really helped me. And also, you get kind of tips on what to do. Like, for example, if they have problems eating, how to thicken the liquids. And you also find out about the different dynamics and how different family members would react to the loved one being sick. And then also, you learn about the self-care, because you do need to take care of yourself as the caregiver. I mean, my dad was a primary caregiver, but still you need to take care of yourself. And also, like, one thing, I've done support groups up until both my mom and my grandmother passed away. And then I did take a break from FTD probably for about 3 years before I found an observation study at UPenn, just because mentally I just needed to step away when my grandmother passed in 2013. So I just stepped away from the community, because you do need to sometimes if you can't make it your entire life. So that is.
Well said.
So I would say, like, if you have a loved one, just go ahead and remember to take breaks and take care of yourself too.
Yeah. All right, next question. And well said, everyone. Can you explain in basic terms what would make the trial successful, and how would that be measured? Carmela, do you want to take that one, and I can support if needed?
Sure.
Probably not needed.
So
Yeah.
when you design a trial for a drug, you have in mind what effect it will have, right? That's the whole point of doing it. And then you have to measure that effect. So, although, everybody wants to cure a disease, right? And just eradicate it. But actually, if you think about it, there are very few cancer drugs that even can do that. So what we have in mind when we design the trial is, okay, it's going to help with, let's say, behavior and cognition. And so then you measure that in some way, and you make that what we call your primary endpoint. So it's really important to remember that trials are designed to be able to show that it's efficacious to help somebody do better with the drug than without the drug. And so you have to choose what the primary endpoint is. And of course, those are crude, right? Because you're trying to measure something that is in the brain, physiological, but we're not taking a piece of the brain. We're trying to measure the output, the outcome of that brain change. And so the measure that was chosen was this this scale that we use it was designed for Alzheimer's disease, in all honesty, and it evaluates somebody's ability to function in their daily lives, because I guess, that's a very important outcome, and, how well they're doing cognitively. And so that's, it would have been successful if the difference between the placebo arm and the drug arm was large enough to really be what we call meaningful, clinically significant. So sometimes you can have a change, a difference, but that difference doesn't really translate. At the end of the day, you don't care about those numbers. All you care about is, what has it done for your life, right? How much benefit has it given you? And so that's what it didn't meet. We don't have all the data to show, did it actually help anything else? Did it, what it did? We don't know yet. Well, once the trial is published and out into the public space, we'll know better. But all we know now is that clinical trial scale, the trial, it's called the CDR, Clinical Dementia Rating Scale. It did not meet that endpoint. I hope that explains that.
That's so well said, Carmela. And one of the things that press release did also mention, so as you said, Carmela, it can be difficult to reach that primary endpoint on the clinical measure, right? And you need to see that benefit. They did have progranulin as a co-primary endpoint in this trial, and they did increase progranulin levels in the plasma. But what the regulators, like the FDA, their job is to make sure that something is proven to have benefit and to be safe before it is allowed onto the market. And so they're looking at the whole package of information. And one of the things that they also shared in the press release is they'd also done some additional measurements of, say, fluid biomarkers, which we assume are things like neurofilament and GFAP, which is a measure of inflammation, as well as volumetric MRI. So to show whether or not the actual damage to the brain or the shrinkage of the brain was being slowed. And that was not beneficial. Like, there was no benefit or change that was seen. And so it reinforced everything that Carmela just said about that primary endpoint that people care most about. How is it affecting how somebody functions and feels? That's what Carmela was framing as that primary endpoint. In theory, for diseases like FTD, you can have a change in the progression of disease that might take even longer to show benefit. And that's why we look at these biological markers to see, is it maybe helping at the biological progression? And if we just waited longer, maybe they would have seen that clinical benefit, but there was none. From at least from what the press release says, there was none. And that is why I think what we hear of why Alector didn't move it forward. And Carmela, can you also answer it? What are the primary outcomes of the study, if any?
So the primary outcomes of
I'm sorry, I misspoke. What are the positive outcomes? We just discussed the primary outcomes. What are the positive outcomes?
The positive outcomes is, as Penny has already said, that the trial could be done, right? I think that actually is a really positive outcome, because in rare disease spaces, there's a lot of reluctance for companies to even enter that space, even when they think, oh yeah, I might have something that actually could target. So I think that's the positive outcome. And the rest of them we'll have to see, right? Like, sometimes in trials they'll do subgroup analysis and say, actually it seems to help this group, or no, it didn't do anything for anybody. So that data, I don't know. Right now it was was mostly a press release to tell us were the big goals of the trial met or not.
And we know this is the first step, right? So Alector's released a press release, but we are expecting to see much more detailed, thorough analysis shared with the field, and hopefully data, so that it can truly, I mean, at the end of the day, a trial is not fail if it's a step forward, if we learned from that trial, right? Where it's a failure is when things are not, those insights and that data and that analyses aren't shared to then inform that next shot on goal. In the trial, was Alector regularly collecting cerebrospinal fluid from the trial subjects? As I understand, the answer to that question is no, that they did that in some of the phase 2 trials, but they did not think that there would be enough. They didn't think that the participants in the trial would be willing to go through regular draws of the lumbar punctures to get the cerebrospinal fluid. And so they needed to make sure that the protocol was something that as many people as possible could comply and then continue all the way through. So they did not include that, even though it would have been a better readout for whether or not progranulin levels in the brain were increased. Carmela, Heidi, Dari, does anyone want to change that answer or refine upon it?
No, I would have to say that, I mean, I've only got it at the first year. I would get it like once a year, but then after, or once, I got it a couple times, but once I hit like 6 spinal tap, 5 or 6, UPenn said, we're not going to give you another spinal tap. And I was not pushing for that
So you
at
were the
all.
phase 2, right?
Yeah, it was phase 2. Yeah. So
Yeah.
I don't think they wanted to even continue because I dealt with spinal tap leaks as being super active. So the problem was that I was very prone to leaks and spinal tap headaches.
Right.
So
Which is not everyone, right? So a
yeah,
lot of people, it's not as big a deal. Yeah.
Yeah. Yeah. But the question is very appropriate. We don't know.
Yeah.
You know.
So how effective and reliable is neurofilament light as a biomarker? Carmela, I think that one's up for you as well.
Yeah, I mean, I think everybody, I think neurofilament light is a really good marker of neuroaxonal injury, but it's not very specific. You could have nerve damage and your neurofilament light will go up. In nerve damage, I mean, like from a peripheral nerve.
I
So
heard somebody say you could bang your elbow against the door and your neurofilament light levels
yeah,
might spike. And
if
so
you
what
fall,
is that? Yeah.
hit your head, your
Yeah.
neurofilament light could go up for a few days. So it's not very specific. And so, I mean, I think there was great hope that this could actually predict who was going to convert or who was going to become go from pre-symptomatic to symptomatic. I think that jury is not completely out yet. I think there are a number of other markers that people have been working on. So I think it was rational what they did. But, this trial started many years ago, right? And every day we learn new things. And would they have used neurofilament light today? I'm not sure. If, maybe the levels that they used, I'm not sure. So there's, much progress has been made even during the time of this trial.
Yeah. And it was that elevation of neurofilament light that they were using to try to say, okay, this person has a gene and here's high neurofilament. We think they're going to convert. And that was what I was referencing earlier, that ended up being less predictive or reliable than people thought. And maybe it would have been if it was more multiple repeated measures. I admit I don't know the details of how they did that initially, because somebody could have that spike from that fall that you're mentioning, Carmela, and then maybe it goes back down. We don't know, right? I think we're running out of time for questions, so I think this may be the last. Would it not be valuable to continue to follow these individuals who are taking the drug to track progression? Hopefully Alector will be doing that. I can say, Carmela, do you want to start?
I think one of the things you need to think about is, firstly, there's other things available, right? So maybe somebody would be more interested in going towards a different therapy, and that option has to be available for them. I think in good conscience, a trial shouldn't continue if they really don't think this is going to give people a clinically meaningful result. And like I said, we don't know the specifics of the data. We just know the high level bird's eye view. And I think from that, they felt like this wasn't worthwhile pursuing. But, like somebody had already mentioned, whether it's good, it's worthwhile for asymptomatic, I think we have to see. But they have to act in good conscience.
They do. And they're also, it's a for-profit company, right? And so they don't think this is going to be a treatment that they can bring to market. They don't have the funding then to keep following people forever. I would say that people can also, many folks I know are also participating in natural history studies like all FTD. They can disclose to their research clinicians that they were in the trial. If that was an opportunity, they can also join the FTD Disorders Registry, which is a longitudinal protocol of how somebody's disease might progress over time, as well as a tool to connect people to trials that can emerge over time. So, it's not the same thing. It's not a centralized group, but if there's a true outlier, maybe that somebody's really benefiting, maybe it could be something that could emerge. It's tough. But I think with that, I am instructed, we're not done yet, so don't leave everyone, but we wanted to take a few moments to talk through some of the parameters of the broader field. So again, this was a huge emotional hit for everyone, right? If we can get the slides back. The Alector trial, despite the results, it didn't work. But what are the reasons for hope? This Alector trial, it was a major step forward towards FTD treatments. And we want to reiterate our thanks to everybody that was involved in this study. Next slide. There are also a lot of reasons for hope that relate to the momentum, both from what was done with this trial, but also the broader field. So there are a lot of important treatment ideas that are actively in clinical testing for FTD progranulin, but also other types of FTD. So there's a semantic variant FTD trial that's active right now. There's many PSP trials active right now. And there are many other treatment ideas that are getting closer to clinical trials for all types of FTD. There's also new tools that are in development to make these trials faster and easier so that we can get more shots on goal so that we learn more from every single trial. The FTD Disorders Registry was transformed last year in an all-new protocol with a longitudinal study and a lot of different types of resources. And I really can't emphasize enough that the ability to find participants for these trials, like right now we don't know where people are. And it's really hard to convince a company or an investor to launch a program in a space if they don't think that they'll be able to test if the treatment's going to work. The registry is a secure database that protects people's privacy, but figures out where in the world and in the country might people be. And that is a really important tool, both to empower the voice of the community, but also to then help with this clinical trial readiness to, when there is a trial, to rapidly be able to get the word out to families. Bipartisan support for dementia research in the USA, as well as around the world. It has been quite profound, the explosion of science in the last 10, 20 years. And there are many, many truly committed researchers. I know I came to the FTD community 5 years ago. That's how long I've been at AFTD. And it is remarkable to me of how committed so many of the researchers and the clinicians are. And I also want to give a shout out to the NIH renewal of support for all FTD2. It is the longstanding bipartisan support for the NIH that has made this clinical research study possible. And they did just recently renew the support in the midst of a lot of geopolitical change, absolutely. But this clinical research study is continuing and there is bipartisan support for research at the NIH. Next slide. So another reason for hope, and yes, I'm biased, but I also think it's true that AFTD, it's why I work here. Our mission is to improve the quality of life of people affected by FTD disorders and to drive research to a cure. Our work spans support, education, research, advocacy, and awareness. Scientifically, our research team is really diving in deep on this clinical trial readiness because we are committed to having as many shots on goal as possible to bringing effective treatments. Improving access to diagnosis is central to all of this, right? There are new state registries that are emerging. There was one in New York that was just the first ever state registry that was launched. This is completely different than the concept of the FTD Disorders Registry. This is talking about a clinic-based registry of how many people are being diagnosed with FTD. We don't even know those things. It's really been hard to get accurate data on some of these things. There are new, incredibly wonderful advocates like Emma Hemming-Willis that are doing incredible work to expand awareness of FTD, which in turn helps people get diagnosed and get the support that they need, and also find out about the clinical trials that might be right for them. And there's so much more. Next slide. I'm looking at the wrong piece here. AFTD is just one of the committed nonprofits in this space. You guys have heard throughout this meeting about the Bluefield Project, which is a nonprofit dedicated to advancing the development of treatments for FTD, especially progranulin FTD. There's other more recent groups emerging like Cure-GRN, which has been a phenomenal source of interconnection and support for many of the GRN families. And you can see from this slide, there are a lot of nonprofits committed to this space. And I think it's really important to remember that some companies will come and go, right? They get their shot on goal. If the treatment doesn't work, they may not continue in this space, or certainly if they can't test their treatment, they won't stay. But these nonprofits are here to stay, and this longevity provides stability and support for the scientific community, and it continues to provide investment for the families that are affected. All of that combined is going to help get us to the effective treatments that we need. Next slide. So the other reason for hope, in my opinion, is the active, informed, and engaged community. There are so many opportunities for action. So again, please consider joining the FTD Disorders Registry. Look into opportunities to enroll in other trials and research studies. Consider genetic counseling and testing. That is always going to be a personal decision, but particularly for people diagnosed, AFTD does recommend that families consider getting tested, particularly when you're diagnosed. There are other types of FTD research as well that can really help accelerate research, as well as accelerate understanding of how do we make it easier to live with this disease. Things like care partner support and interventions, things like brain donation. People can also join AFTD's efforts to volunteer for community awareness and fundraiser projects. Join AFTD's Advocacy Action Center. That is also a very powerful resource to help advocate for government funding for research, for example, amongst many other types of advocacy initiatives. And share your lived experience with AFTD. In turn, we can help you connect it to give people opportunities to share their lived experience across the space with researchers. Please spread awareness of the resources that are available to other families, to your community networks, the people that you come across. AFTD does have a lot of resources specifically around genetic FTD to help guide families in this really challenging space. And stay informed and share information with your own network. You can also, there's a QR code on the screen that you can scan to subscribe to the AFTD Science Digest. And before we end today, I'd like to ask each of the panelists for their own reasons for being hopeful or their own call for action. I'm going to point first to Jari.
Thank you, Penny. And thank you again for hosting this. Again, I just really want to stress the reason that I am so hopeful about this is that there are still 5 other clinical trials in the progranulin space. I think that is extremely important. I'm also really, really moved by the patient organizations that have been sprung up that are speaking for the voice of the patient and also making sure that there are ways for people to connect. The other thing that I'm really hopeful about this is I've met with a number of the pharma companies. I met with Alector since I'm in the Bay Area, San Francisco Bay Area. I did meet with Alector for AFTD or for FTD Awareness Week, and they had a lot of questions about what it is like to live with FTD, what it is like to care for somebody with FTD. And they really, really were caring people. And that has been my experience with everybody that I have been dealing with in the pharma realm. They really do care about what they are doing. Even though they are private industries, they are focused on trying to cure this disease. And that gives me a lot of hope for the future. Thank you.
Thank you, Jari. Carmela, I'm going to point next to you.
Yeah, I think, we should remain optimistic. I think they, Alector accomplished something that many people actually thought was never going to happen. Rare diseases often fall prey to the fear of not being able to do a trial, find enough people. They have made so many connections around the world. I think at the end they were like 66
trial sites at one point, yeah.
Yeah, 66 trial sites. Those people now, they're a network, right? They're an asset for the whole FTD community. So I think, you know, we should remain optimistic. And I think you know, it took Alzheimer's a long time. I don't think it's going to take that much time for FTD progranulin because there's been so much investment. There's a lot of companies already in that space, many more than we would have really expected. So I think we should really remain optimistic and yeah.
Thank you. Heidi, what would you like to add?
I would like to add, yeah, no, I definitely feel there's a lot more hope. One of the reasons why I had done the research originally is because my family, watching my mom and my grandmother both have it. And I actually went on Facebook and looked up family members and cousins that could be impacted by the gene and made a list of about 20 people, counting second cousins, third cousins, aunts, uncles, how much this clinical trial, even though it did not make it to FDA approval, has benefited the future, not just for family members, but my nieces and nephews also won't have to worry about it. I do feel there is about like a 99% chance I'm not going to have to worry about a FTD like my mom did. I know that there's no cure or treatment, but I do see the advancement in medical treatment. And as I mentioned earlier, I am a contract worker for NIH. I do see how clinical trials improve daily with technology. And especially, I think there's a lot of hope with the gene therapy studies too. So
Thank you, Heidi. All right, that wraps up our town hall for today. Thank you again to our guests. Although we were not able to address everyone's questions, remember, you are not alone. The AFTD helpline is another great way to get the advice, support, and resources that you need. Thank you, everyone, for joining me. And thank you, everyone, for joining to listen in. Take care.
Thank you.